Yeah, I think we can get started by updating the toolbox. So we are just waiting for more people to just come in. So nowadays, the updating will just within this application. So I have current version 3.5. So if you have this, you should be able to use this menu to check the updates. Then just allow this update to install the new version. Then you just restart, then it should be okay to the latest version. So I get rid of this command line, and now the application is installed in the default package folder, and we can then add an app. So on my screen, so I just update to the latest version, then I just restart the interface. So now the version is 4.3. 4.3, yes, should be the latest version. Okay. You can also now see, okay, I can install app. So this just put a shortcut in the app here. So after you click that button, you should be able to see, okay, there's a app called Single Cell 2. So you can put a favorite. So you only need to do this once. So this will make your life a little bit easier. You just click this button, and every time you don't even have to type this command line here. So if you come from here, and you can just click this button and to run this application. Okay, just go around and make sure you have this updated version installed. Then we can start today's program. Again, what you need to do is just check the update and let the program update itself. Then you install this application, so you can have this orange button in front of you on the top, so you can just click next time to bring up this application. So this is some recent update. I have made this feature available for people. You check, yes, it should be in there if you drop down this right here. So
start it.
Yeah, you can start it. You can unstart something else.
Yeah.
You got it? Yeah, so because you are using the old version, so this is not working.
So I checked for update.
Yeah, so you check update, and so you see, yes, nothing happened. Oh, okay. So because you're too old, this version. So what you can do is to yeah, last time I said delete the whole thing if you can. Yeah, then let me remove from parse. delete the whole thing because you want and then you want to add as an app. Let's see, as an add-on, you search SCGEA toolbox. Then you install from here. That's the latest version. Then from now on, you can update within the program. Yeah. Okay. So now it says you're installed. It seems you already have this. So can you run this? Yeah. So you have the. Yeah, but now you update from here. Yeah, so you need to delete the old version. So now it's just run from here. Okay, yeah, good. Yeah, 4.3 today, yeah. So 4.3 today, yes, the latest version. So which version you have using right now?
I don't have that option to install.
Yeah, so you check update, then you update from the. Let me see.
25.3.
Yeah, so this should be the latest version. Yeah, you need to restart because it was installing. Yeah. Now you have the latest version. Yeah, 4.3. Yeah, so add the. Yeah, so it should be in the on the top, the blue one. No, on the left. Yeah, the orange one. Yeah, so you set up. Good. So what we are going to do today is in this channel post, and we will do the HVG analysis, and also I will show you the spleen DV method to compare two samples when you have different highly variable genes. So this is from last week. We have regression and found the genes which jump away from the regression curve. To see those genes are the most important ones. So this part. Then we will look at SVD function. Okay, this is the built-in function. Using a small example to get familiar with what's U, S, what S, what is V. And then we use this example matrix to do SVD decomposition. So this example is from the literature slide page. And then we will look at this use SVD or to do this low rank approximation using image to see how we can extract the most essential components from an image, and then use those essential ones to reconstruct the image. You only select those most important ones, so give you effects of a denoise, and if you have a very clear picture, then what you get is a compressed version. It's very similar to the original picture, but much smaller in terms of size. At the end, we will play with example with ICA to do this independent source components analysis. Okay, so let's start with the highly variable gene expression analysis. So the example I'm going to use is recently published paper from my lab. So let's search for I hope we can find the paper. We need to find the paper by looking at the spleen DV. So this is the keyword of method we developed to allow you to compare highly variable genes cross samples. Okay. So this is a paper discuss about this application, this method, and the 3 case studies we used to show, okay, this is really work. So what you need to do is to browse this page down. You don't have time to read details, but the illustration of the idea. So we will build a 2 regression curve using spleen method. So this is not a real regression because we are not construct analytical solution of this curve. We just follow the majority of the genes and build a curve, go through it. And we will build 2 curves, one for blue sample and one for red sample. Then for the same gene, because you have 2 samples, every gene you have 2 points. One is response correlated with a blue sample, another from a red sample. We want to see where this gene is located respect to red and blue curve. If their location is very different, then we see, okay, these 2 show different variability regarding to the rest of the genes in the same sample. So we will find those genes. Those genes called genes show highly differential variability. So you compare 2 samples. So the first case study, we used a mouse adipocyte. Another case study is this is also the mouse study in the liver fibrosis. And the third study is colonal cancer research. It's from a human, maybe. Yes. Okay, what we need to get is a set of this geo number, okay? So if you want to study the first case, you need to have this 2 sample ID from a geo database. And also this. So what we can do is just copy this ID and to do a search to lead you to this page. So this page is where this sample is collected. So if you look at this as ID, this is the sample we will download. We will download them and put into the MATLAB to compare with another sample. So this sample is from liver tissue and 6-week. So you have to read this. So where they come from this study. So another ID is as a pair is this. So 5, 6, 2, 3 is the different sample. Yep.
So we download 3 files for the first sample?
Not now. We can have a shortcut to download them. Okay.
Oh.
So I just show the samples right now. So you can put these two records side by side and see the difference between them. So this is a healthy liver tissue, and the ID is 5623, and this 5625 is another sample which have some problems maybe. Okay, so we are going to download these two samples together to the MATLAB. So we need to get the ID, okay, copy the ID, come back to this page. And you select this geo accession number, okay. In here, you will type two of them. One is 23, separate with a comma, and then 25, because these are two samples we selected. Let me give you the, please. Say that again. Let me zoom that. So magnitude. Yeah, so this is two ID we are going to use as an example. Yeah, so if you have this, you can start to click OK, and let it to download and merge the data together. So this is the way you don't have to do independently, one by one. Just let the program to run it. Yeah, go to the latest version. Oh, the problem. Yeah, so it's a network problem. So let me just try that again. Do you have this ID? I do. Yeah, do that again. Mm-hmm. Yeah, so this is a network issue, and recently not so stable. Yeah. If you have trouble, and you can just download the one I prepared for you. My machine, and see whether it can go through. NCBI net database sometimes not so stable recently. You got a problem? So if everything goes through, you should have the annotated the results. If you don't want to bother to do this yourself, you can download here. So this is the file I prepared. Oh, that's a different one, but it's okay. Yeah. So the one I prepared is for this one. So the last example. Okay. So you can download the data which already processed for you for the case study 3. So you don't have to wait here, or you have issues with connections. So on my screen, the second data set is being downloading right now. I hope it goes through, and so we can have something to play with. So if you don't want to wait, or your computer is always constantly give you the error message, you should be able to download this processed sample, save into your hard drive somewhere in the download folder maybe, and then come back to this dialog window to see, okay, I want to import a MAT file, which is the first option in the menu. You should have that processed data for you. The example on my screen right now is finished downloading, and it start to ask me whether I want to process this. I will say yes, this is mouse data. So the program is working on dimensionality reduction, cluster cells, and annotation. Yeah, still waiting, yes. You have, okay, you import data or you download?
The
Download one, okay.
the imported one.
Okay, okay, good. Yeah, you just wait. What's this? Species, mouse, yeah. Okay. So this analysis is cell type specific. So we have to decide which cell type we are going to focus on. You cannot mix different cell type to do this highly variable gene analysis. You need to have a relatively homogeneous population of cells. Okay, so this is my result here. You should be able to see this separated two group of cells, 23, 25. So this was just sample ID. If you look at the cell type, I believe they're fibroblasts versus stellar cells. Let's see. So I'm reviewing the cell type in there. What I can do is to look at multi-grouping batch ID with the cell type to decide which cell type we are going to focus on. So if we decide to do with this blue fibroblast, you will have this group against this group, which that's between two samples. I think that's enough to do the analysis. So I'm going to extract fibroblasts to compare between these two samples, okay? I hope you understand these two pictures are the same group of cells, but they're just labeled in a different color. And those cells have been annotated as a stellate cell. But this group is from one sample, and this group is from another sample. So in this picture, we have two samples, and the two cell types. So each sample, so this sample contains fibroblast and stellate cell. There's also fibroblast stellate cell. So we need to decide. We are going to do fibroblast, okay, the DV analysis. Yeah. Different samples I will show you in a separate view and what we have. Yeah. To get this, you go the big eyes here. Yes. Then select multi-grouping. Then OK. Yeah, you see the same picture. Yeah, similar idea. So you have to decide, say, which cell type you want to compare. Just use this. Yeah, if you do this, the next one. Yeah, and then click on one of the cell type, maybe the blue one, point to here. Neurons. What's the blue ones? Yeah, you have to select which one you compare. So this is from you download from the one from the channel. Yes. Okay. Okay. So I show you this example side by side with this. Yeah, so let's see which cell type we should use. I don't know the example. Yeah, what we have here.
Should I do it by size?
Let me load this example in there so we can have. So this is a case study number 2. I'm going to load case study number 3 as I prepared here. So I download this and open another. And load that example in here so we can do a quick decision on which cell type we are going to use. So this is a more complicated sample. There's human data, more cell types in there. And we have to look at the groups. Okay, looks like we have group, this group compared to that. So this is 8-3. Is this correct? So intersites. Yeah, so I think in this analysis, we are going to compare intersites. So we will extract intersites out of this sample to compare red with blue samples. And those are irrelevant. Those are some of these maybe cancer cells. I don't know. So we are going to compare this yellow with that yellow, okay? So for this example, we are going to see, okay, I'm going to extract cell by group, okay? So it's this button here, and then you check the cell type. Look at alphabetical order, so we look for the enter sites right here. If you are using the different samples, we will play with this later, okay? So now it's enter sites for this sample, and you select. So still working on this. So we are going to extract this yellow group of cell out. So this will be 2 samples. The cell type are intersites only, okay? So this is a very simple setup right now. We can close this because we already decided to compare these 2 groups for their intersites relationship there. And come back to this sample. For this sample, we are going to compare fibroblasts, okay? So we extract fibroblasts out.
When I sorted the batch ID, it doesn't come off, and mine doesn't look as distinct as yours. But these are the.
Yeah. Yeah.
Should I
So
select both?
you don't have to select. You already have.
Okay.
Yeah.
So
So this the cell type. What cell type this is?
enterocytes.
Are you sure you're using the example download?
Yeah, the one from Teams.
Oh, from Teams. Okay. Yeah.
This one, right?
Yeah. Good. Yeah.
How can I double make sure that? Should I just?
Yeah. You can check what cell type in there.
This one?
Yeah. By label the cell type. Can you rotate the C2 samples? Click this button here. Okay.
Oh.
Yeah. So you got it.
Thank you.
Yeah. So if you are working on the fibroblast examples, we can extract fibroblast in the same way. To select this, to select fibroblast, and click select. Okay, then you can close the old, because, so on my screen I have 2 examples. This is a compare between intersites. Okay, if you use the example from the channel you download, let's compare these 2 samples. Well, this side is the fibroblast example. We only have fibroblast. This fibroblast is between 2 samples, okay? So we need to have this clear setup, and we know what cell type we're focusing on and what 2 groups are, okay? The numbers of cells are comparable, so we can estimate the variability relatively accurate. So it depends on which example we are using. Now we are doing the same differential variability analysis. Okay, so this will let's see, this is some human data. Yeah, so you will experience a dialogue like this. So we are going to remove some less informative genes to make the results more meaningful. Then you select compare these 2 groups. Yeah, this is difficult. Let me try this. This is mouse data, and remove those. Compare two samples. Using splend method. If you are using human data as an example, so just don't remove those genes, otherwise you don't have enough cells to play with. So if you are using the mouse data here, and you will eventually get a picture like this. This is two curves and with the genes. So I show you how to, if you do this, you don't remove human, you don't remove those. You just see no, something like that, to keep enough genes to be in there. Because the number of cells to fills still. Yeah, so there's some problem with this human example. What I can do, I can just upload this to the channel, so if you want to download, you can have that. So this could be the better example. It contains more cells to allow you to play with. Okay. So this will allow you to start to check genes. If you found, say, this gene, the relative position with these two samples. So for this gene, there's no much change. The locations are very similar because their profile look similar in these two samples. So we only consider this as a gene which have a significant difference in their variability change. Then we can try different samples, this, that. So you can click basically to find which gene will be differed very much. You can get from the list. So this will be the top one. We show the most difference. I try second one. So this gene, for the orange sample, it looks like this, and this have very low variability because the expression level is also low. So if you look at this gene, same situation. So those are the top genes have been selected for this example. So this gene list is a result of the DV analysis, differential variability. you can have this. Yeah, yeah, just do this. Just a table, yes. Mm-hmm. Okay. Okay, yeah. So this panel shows the expression profile across cells. Oh, you're always there, almost there. Right? Okay. 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 Mm-hmm. Yeah, so this may be not enough. We don't have enough cells to play. So what you can do is just download a new one. Download the one I just prepared in the channel. Do you have this?
Yeah.
Yeah.
Well, I have this one.
You download this one already in there?
I do have it.
Okay, you download. Okay.
These two, yeah.
Yeah, it's here. This one here.
Okay.
So you can just run analysis DV with these two samples. Small state. And then you click through, you should have this picture. Mm-hmm. Batch ID. Workspace. Mm-hmm. Okay. Okay, yes. Okay, yeah. Then you can start to click on any of the genes. Maybe, yeah, you should find a difference in the two samples. Yeah, so that's the idea. And the list of genes you can find from this drop down this menu. Yeah. Mm-hmm. Those top ones, yeah. Okay, I think we can finish off this part. So those are the genes. If you are interested in some of this, you want to check what the gene expression change. For example, this gene, there's no change. They're all on the same position, same curve, location. So this is not interesting genes for us. But if you found different ones, maybe have a different location. So these genes are exactly the same almost, right? Their location, both on the curve, similar level of expression, average variability, and follow the major pattern. So this gene definitely have nothing to do with this comparison of this sample. Those are the top genes, and you may want to check them one by one. So this when 4 genes expression level higher, and also the variability increased. So this most likely to have some function implications to make the orange sample different from the blue sample. So that's just a way to interpret. So some of this pattern was driven by the average difference. If you want to check the gene function or information related to this gene, you click this button to visit this page. So this is for human genes. Okay, I think we are done with this part of the differential variability analysis. Just remember you need to compare same cell type. Okay, you don't want to compare this sample cell type 1 with that sample cell type 2. It has to be comparable samples in order to build the curve make sense. So now we are going to move to the second topics in our agenda. So I can close all this, clear my screen, close all. So I'm making a small example matrix. SVD function to decompose it into 3 matrices. So U, S, V, okay? This is SVD function to decompose this. So let's look at the dimension, the size of U. So this should be a 2 by 2, okay? So S is a 2 by 3, and the V is a 3 by 3. So you get the idea about the dimension of these two, okay? The reason is because A is a 2 by 3 matrix. The original one, we make a decomposition. So decomposition also is a factorization. So that means we should be able to do the multiplication and to reconstruct A. It's not correct. The reason we need to do this transposition is because the V generated by the SVD function has been transposed. So we need to transpose back to its original form in order to make the product of these 3 matrices and reconstruct the original sample. Yeah.
Go back to the previous entry. Just one line above.
Yeah. It's this. So we are doing a process which decomposes a matrix, then put them back. So in this case, it's 100% recovery. Okay, no loss of information. So what we can try is this minus U times S times V. Okay, so this expression gives us So you see this all the time in the expression of this. So if you have original one, you want to compare the reconstructed one. So you have this reconstructed one, you want to compare how different they are. You get some idea. So you always see their norm. So f squared, something like this, you always see. But this part of the reconstruct, we use u times sigma times v to approximate it. And then we want to see, okay, what's the difference between them. So we want to make this small because we want to, the reconstructed version is similar or close or exactly as the original. So that's we want to numerically check this, make sure they are right. So the expression of this is what I'm writing in there, the function. Okay. I believe we can use this. Okay. So this number is really small. So it is 10 to the minus 15. So close to 0. So this tells you the difference between this and its reconstructed version. Although it's matrix, there are some precision problem, not 100%, but the difference is really, really small. Yeah. This one here. Yeah, so this is called the Fabrician. One of the options you can do the norm. This by element, you add 6 elements together, and this make a small values. Otherwise, the norm is for the matrix itself. It's computed differently. If you don't have that, you should have a very close result, but the way to compute is different. Let's consider the eigen, and also, so if you, so the norm of the matrix, you can do read the documentation of the norm function and see how it was computed. It involves some of this eigen decomposition. Well, this version is the most straightforward. It's just add up the difference values. As I said, this SVD is very stable, it's applicable to all different kinds of matrix. So our example was 1, 2, 3, 4, 5, 6, simple one, but I make a more complicated one by random number. So this matrix is 5 by 7, but for SVD, it's just the same. So the results is also very small. So basically, you can reconstruct a completely different matrix input matrix to decompose it, and also reconstruct the whole thing easily. But you need to know the difference. And in this case, you get U matrix is 5 by 5, S is 5 by 7, and V is 7 by 7, right? That's because your input contains more dimensions, and you need to have this rotation matrix, and also the singular value contains more elements in there. And the original matrix A is a random number generator-made matrix. So the elements, the values in there is purely random. There's no structure in terms of rows, columns. So what you observed from the singular values is they all very similar to each other. Each just contribute a little. Because original A has no structure. You don't have those major components to be learned. Otherwise, you may have a 10 here, 0.5 here, and almost 0 to the rest. That tells you the first element components can explain a lot of the structures in the A. Okay? In our case, purely random. So all the values are the small, all the dimension contribute equally almost. So I think what I can do is to make this, to manually introduce some structure in this A. Now it's purely random, but if I see, okay, I want to make this, see in A, if I select 1, 2, 3, and 1, 2, 3. Copy this. So for all this, I just add values. Okay, 5. Okay, what I'm doing here is I select this 1, 2, 3, 1, 2, 3, this corner here, by adding a constant values 5. I just artificially introduce some structure. So now this matrix is not random anymore. Now I do this as VIDI again on this non-random matrix. Now I want to see the change of singular value, okay? So now you can see the difference about this decomposition and the singular value, absolute value. So this first component become 15 compared to the one which purely random one. So this number tells you this from that direction. There's some of this structure you can learn from this purely randomly constructed but manually changed matrix structure. So if you imagine this as a design for a bridge or building, you have a lot of components linked, then you can learn from there and which direction its major components they linked or form a non-random structure. So that's engineering applications. In the video we see the bridge, under the perturbation start to shake, and you need to find which direction the major change should be, or if you have a matrix like that. So This is just an example, and you can increase the singular values for the first element by make the matrix non-random. Okay, I think it's time for a 5 minutes break. Okay. Okay, I want to use this time to mention the homework. Among all of you, only one student got 10 points, and the sentence I want to read is in my feedback I gave to you. The sentence I want to read is, you have to know this algorithm was not used in its original purpose. The original purpose for the classification is to say, okay, I can use those genes to classify the patient out from the non-patient clearly, as clear as possible. But this algorithm was used in the opposite way. They want to find which cell type they can make this prediction more accurate. So I want you to have this idea to do the opposite, or even from different angles, to utilize a pre-designed algorithm, but applied to your customized problems in a different way. So you cannot achieve this answer using language model, so that's now you get a full score, rather only one student figured out. So the whole idea is just make you think of anything you learned from this course, and you know how to do the classification, dimensionality reduction, you know how to do the regression. Please find a way to think sometimes differently and to make a change. Maybe just change the narrative to apply to your own research. You don't have to be an expert in the data science to make this change if you know the principles and ask the right question from different angles. Okay, you probably can do this. So in our case, we decompose a matrix, any given matrix, use as VIDI. Now we see, okay, if we check the singular values, we know, okay, component number 5 contribute almost can be ignored. Okay, this is a neglectable contribution, so we probably can remove this component 5, only use the top 4 to reconstruct the original matrix. Our original matrix has been randomly generated and modified manually. So we can use image SC to visualize the values of this matrix. So I put the value of matrix, and with this image SC, SC stands for pseudo color, and it's rescale the numbers here and then make larger value yellow and the small value blue, depends on the color map you use. You can click this to give the scale the color bar. But this is a very useful tool to let you to visualize the potential pattern in a matrix. So the color bar is here, okay? So indicate some values reach almost 6, and a lot of this still 0 or negative. So if you have done this decomposition, you know the reconstructed version will be very similar to this. If you do a mu figure, and the U times S times V transpose. I have two figures on the screen, and the figure 1 is original matrix, and the figure 2 is reconstructed. No difference Now I want to use the top 4 components only to reconstruct this, rather than use the original SVD. So let's set up this U with dimension small u. Okay, this is a small version of u, but it's also only use top 4, 1 to 4 matrix. So this is original U, and this is smaller version of U. For the S, we will make this into S4 has to be the top 4. I don't know. We still need, made the wrong answer because we still need, so U4 will be, sorry about this. So we still have to have 5. Let me see. We still need the top 5. So you only need to, so we reduce the number of components, but we still have to keep 5 samples, because at the end we will construct the matrix with 5 rows. So S4, so this is a small version of S, will be 4, 1 to 4, 4 rows, and has to be 7 columns in order to make this product. Okay. S4. I hope this works. So I reconstruct a small version of the A with 4 components. I hope you know what I'm doing. So I use a truncated version of this rather than the original with 5 by 5, but I only use the top 4 to reconstruct a same original. Okay. So if you do the norm between these two, so this is fully reconstructed, you get a very small value. But if you compare with this A4, that's only use 4 elements to reconstruct it, you get a slightly larger difference. Okay. So this is a compare 2 matrices, see how similar they are. So if you have all the elements equal to the original, you should have very small norm. But now I compare the small a4, this variable is reconstructed matrix, but not use the full decomposed matrix to reconstruct it, but use only 4 of them. 4 of them is from the top 4, which is this part. And I remove one of these components, number 5. So the reconstructed ones slightly different from the original one because the total difference is higher in this case. We can still visualize this using A4, using this image to look at this compared to the original. They're still very similar, okay? Because although I use only 4 dimensions to reconstruct the original matrix, but because I select those important ones, so I still have the total recovery quite similar with the original one. All right. So we will further illustrate this point using, let's jump directly to this, so number 5. So I'm going to use this example again to illustrate the points which we can use low rank approximation to approximate the original picture. So here is a picture which and reconstructed. So now we are using a real picture here. What we can do is to copy this piece of code and paste into MATLAB. So we can close this. Okay. Now we paste this example. We should have this image, original image in the IMShow window. So this is the function to show this image as a matrix. Okay. You also should see the change. So this is a 480 by 640 pixel image, black and white. Okay. So your A matrix becomes this. So we are going to use SVD to compress. So the function we are going to use is SVD sketch. So this is a new function to help you to select the most important components, just like what we did. We select top 4 by set up a cutoff and make a smaller USV for reconstruction. So this function automatically generate skinny version of the USV. So if you use standard SVD function to do this decomposition, you will get a huge matrix U, huge matrix S, huge matrix V, because standard full version of a decomposition will depends on your input. Your input is 480 by the rows and the 640 by the rows. So U and V will be a square matrix by that dimension. But this sketchy function will automatically help you to extract the most important ones. Only use those for reconstruction purpose. So if you run this function, okay, let's come back. Okay, I can move on. So this sketchy function help you to find the most important components and only use 288 rather than the full spectrum. So standard SVD will give you 480 by 480. But this sketchy function found, okay, you only need 288 rather than 480 to reconstruct the original. So you can see this is a reconstructed version. No much difference from the original. So that's the point. Let me bring up this page again. Original and reconstructed using 288. Now, this time we change this value, this cutoff. It was 0.01. Now we use a larger cutoff to truncate more less important components. So this will give you only 48 dimension to reconstruct the original picture. Now it's getting blur. The reason is we only use 48 components to reconstruct the original matrix. So this makes the image size much smaller than before. But the cost is we lost some original resolution. Okay, so you can also see I want to have the limited dimensions. So if you give this, you will only keep top 15 components. if you keep top 15 components, you will certainly get even blur picture. Okay, so that's what you get from the 15 components. Okay, depends on your application, what level you want to compress this picture. So you can replace the original matrix with 3 smaller matrices. And to let the matrix multiplications to reconstruct the original matrix as a picture. All right, now your job is to look at this matrix, type this in the workspace, the command line, and to do the decomposition to find the singular value of this. What is the first one, how big it is, what is the second one, do we need a third one, something like that. So that's your input matrix, okay? So this is the example used in the slide, in the literature slide. Yeah, for this example, we have 7 samples, only need 2 components to reconstruct the whole thing or approximate it. What you can do is do times S times V transpose. So this will give you the a approximation, should be very similar to A, okay? Those are very small numbers, close to 0. Okay, now we are going to play with the last example to show the ICA function. So let's download this zip file, and also we need to find this file in the download folder, and right click, and you have to extract into a real folder. Not a zipped, but this is a real folder inside the download. There are PCA underscore ICA, and also one more layer, and the 2 into this folder. Now from here, we are going to switch. We are going to change our folder, current folder, to here. Okay, make sure your CD, you change folder to this location. Yeah, just make sure this is your working folder so we can have access to all the functions in this folder. Let's see, we should.
One, two.
Yeah, just I want to have a better control for the volume here before I play this. So this is the file which inside the audio file, and so this is a demonstration. Okay, so I play the
3, 4, 5, 6, 7, 8, 9.
So this is a mixture number 2. So that's the 2 signals.
1, 2, 3, 4, 5, 6, 7.
So these 2 signals are mixed of this people read 1, 2, 3, 4, and with the background noise of the music. And we have 2 sources. The mix 1 is I just played, and mix 2 I played. So now if I do do the decomposition. I think let's play this example. So this is the original source, the true one, and number true two.
Long list of American fast foods that have successfully made the long march to Asia, from Seoul.
So this is the second, and the third is this. Now we mix the three together, okay?
Long list of American fast foods that have successfully made the long march to Asia, from Seoul.
This is one mixture. Okay, you need a second, you need a three mixture in order to decompose, okay? There's a second mix and the third mix. So within this three mixture, different salts have a different strengths, okay? But you learn the similarity and the difference between them and reconstruct this mixture matrix so we can decompose it. So let's see this fast ICA decompose results. Okay, and then the second, this is just from a mix. Then
long list of American fast food
to separate those three sources, right? And if you use different algorithm, and this did a job, another,
long list of American fast foods that have success.
and this is a different ICA algorithm.
Long list of American fast foods that have.
So this algorithm,
Long list of American fast foods that.
as you can see, it's not, you can hear, not as good as the fast ICA algorithm.
Long list of American fast foods.
It's not separate the three sources very clearly. It still have some level of mixture. So this is like a three source mixed example, and the one I just showed you, this is a two mixed, okay? It's a two source
1, 2,
mixed,
1, 2,
and then we try to reconstruct the first one,
1, 2, 3, 4,
the second,
1, 2, 3, 4. 1, 2, 3.
I don't know exactly what they are doing.
1, 2, 3. 1, 2, 3.
Okay, so this is a two source mixture, but the decompose maybe into three sources.
1, 2. 1, 2, 3. 1, 2.
Yeah, so we don't have to go to the details each individual function, but you get the idea about this true signal, three different observed mixture and independent component, okay, separation. All right, I don't know if you can play on your machine. You don't have a speaker. Nothing. Yeah, so you do need a speaker to make this work. Do you, I don't know whether this is required. I don't think so. It's pop up on your machine. Okay, so maybe one of the functions, some of the smooth function require signal processing toolbox. Yeah, MATLAB is really very strong package for doing this signaling processing. Have the function built in. Yeah. I think that's all for this week. Let me know if you have any questions. Otherwise, see you next Tuesday. Next Tuesday, we will discuss the pseudo-time analysis. Okay, follow the trajectory to study the cell change from one stage to another. We will use some regression, some of the dimensionality reduction. All right.